Hold on, just okay. So everyone can see my screen.
Yes.
The presentation screen. Okay. Hello, everybody. I'm Chris Hellriegel. Today we're going to take a look into the AI sample finder. The AI sample finder is artificial intelligence sample finder. It is a device that is attached to the microscope that rushes up, gets you up to speed in no time when preparing the sample, when placing the sample, loading the sample into the holders and placing the sample in place. It will generate an overview to get you a jump start to get to the imaging part quicker. Just as a quick disclaimer, the AI sample finder is not an automatic acquisition kind of device. It is a device that helps you to get to the sample quicker, to get to an overview, to be able to navigate the sample. It's not a tool to start the actual measurement of the sample. It helps setting it up, but it does not perform it. The setting up of the measurement and the areas still has to be done using the navigator. I'm going to switch to one of the instruments that has a sample loaded to demonstrate the use of the AI sample finder and also use that opportunity to go into the sample acquisition proper and maybe refresh a few items on the sample navigator in order to find the sample, to set up a tile region and to determine what focus spots will be used, but we will cross that bridge when we get there. So first of all, then oh, I'm not sure I'm sharing the right screen. My sharing, let me see here, sharing screen 2. Okay. So without the AI sample finder, when we are at one of the confocal microscopes, we place the sample into the sample holder, we place the oil there, we place the sample more or less to a place where we think the sample is. Using the ocular, we will then search for the object of interest, for example, using the DAPI signal to find out where the nuclei are, or looking at cell agglomerations or something like that, something that lets us understand that we are actually looking at the sample and not some spurious element, a piece of lint or dust that is lying around in the sample. Then we would typically go and adjust the tile and position navigator by moving the sample and acquiring an overview. And then we would set up the regions and the acquisition settings with the focus strategy and so on. So all of this setup is very manual. And to generate this overview can be quite time consuming in order to get to the sample. The sample finding with the AI sample finder uses a full sample camera. We will see that in a moment, how that looks like. It's an additional device. Think of it as a special illuminator and a special camera that are attached to the condenser of the microscope, and that will generate an overview image and will detect where the sample is and inject that directly into the tile and position navigator. So setting up the regions and the acquisition settings, for example, using the focus strategy and so on, then goes on as usual. So what the sample finder does, it gets you up to speed quickly, but it does not perform miracles. That being said, I think before we go to the machine, Zeiss has actually made a quite interesting video that I would like to share with you about how the AI sample finder actually works. So without any.
How
How often have you looked at your microscopy slide, being unable to locate your sample because it is translucent or simply too small? I would like to show you a new way to accelerate your experiment startup. Let's click Find Sample and see how fast this process really is. Locating your sample is as easy as placing it under the microscope and letting the software do the work. AI Sample Finder detects the sample carrier and identifies the sample areas. Every area is focused automatically, and you can start your experiment right away. And now, let's see what actually happens. So, first things first, you don't need to worry about your condenser or incubation equipment getting in the way, because everything is already taken care of. Artificial intelligence will first detect the sample focus, and a low contrast image of your sample will appear within seconds. Then, dark field composite contrast imaging creates a high contrast image of your sample without the need for additional staining. Artificial intelligence will automatically detect the type of sample carrier and cover slip used and locate your sample within the cover slip area. Regions of interest are created and can be edited if necessary. And what's best, AI distinguishes between dirt and your sample. All you have to do is click on Finish, and Sample Finder will automatically determine the focus on every single region of interest. Effectively, Sample Finder speeds up the image acquisition up to 10 times, while being more gentle and less phototoxic than conventional methods. And that's it. Now you will always have the full sample information. You can easily navigate regions of interest, you can use fluorescence, zoom in, and examine regions of interest with higher detail.
Okay, so that is what. So can you all hear me? I would, just to let us know if the video displayed properly, if everybody heard what was in there.
It was good.
Okay, then moving on. What we saw on this video is what we will see then later live on the machine. Just to go back to the individual steps, here we see this all in a little bit more detail. So on the objective revolver, there is installed a mirror with an overview camera and that LED array that is placed on the condenser. You can see it here in that middle image. That illuminates the sample at various different angles, and then the composite image is then reconstructed. So the workflow is it will first try to find a focus, then it will do this dark field composite contrast image, that the one you see on the right. Then it will try to recognize what kind of carrier you're using, if it's a well plate or if it is a cover slide, and this is that blue frame here around the big sample. Then it tries to identify the samples. Here you have many different cuts of a sample, and then it will get you in the ballpark by using a long-range autofocus. So again, here in terms of images, you get an overview image, which is generated rather quickly. It gives you an overview image. It's ideal for remote operation. And there are some imaging modes that I can show you. For example, this coherence contrast method as an all-round contrast for most sample types. It is not high resolution, but it gives you a high contrast, which helps you finding the specimen. And this here is our very own LSM 900 here at the HCBI. You can see it here in action. This is how it looks like. This is actually the sample that is in there is exactly the sample that we see there. And yeah, and this is how the sample finder operation looks like. And without any further ado, I would like to move to the instrument. So we go here. Okay, the instrument is there, and it was just turned on, and I just placed the sample on the sample holder. And this is the look and feel when you turn the machine on. You're in the locate tab. You can take a look with the eyepiece, but since we are remote, I cannot take a look with the eyepiece. So I go straight into this acquisition tab. And here is where you find the AI sample finder button. Let's go there. Let's go into the AI sample finder, and I would load the sample here on the right side. It will now move the stage like we saw in the video before to the lower right corner. I would place the sample in there, and then I go to find sample. Now it's going to move the sample into the field of view. It's a pity we can't see this right now, but it's moving the sample into the field of view, and it's generating an overview image. And we will see in a moment what this overview looks like. It's as you can see here, this is the low contrast image. It's a cover slide with a piece of a mouse, a thin slice of a mouse, and yeah, that's the reconstructed high composite, high contrast image. And it's now trying to detect the sample carrier and the sample. It won't always find the sample right away, especially this one, which is quite big, but in this case, it actually worked. In case it does not work, there is always the option to use the drawing elements here. For example, you could draw an additional sample, an additional rectangle here and there, but it recognized actually quite well the shape of the cover slide and the whereabouts of the sample over here. And the yellow regions can be edited. And yeah, I would just move on to the next point. Now that it knows where the sample is, I go, okay, let's go next. And it's asking me to proceed with a long-range autofocus. The answer to this is yes. I would like to proceed with a long-range autofocus. That takes a little while. Okay. Now it's going to ask us to move the camera to a different area here, for example. I can zoom in. I'm doing this with a scrolling wheel of the mouse. This is the snout of that animal. And I'm going to use this coherence contrast method here to take a look at this. And the typical value for that is something like 0.1. And if I create here, oops, what's happening there? Oh yeah, I selected the wrong one. Yeah, there it is. This here should be 0.1. And that is, I don't know if this displays well, but that is that coherence contrast image that we can see over there. I can move this to different areas of the sample just to check, you know, this is the cerebellum here, just to check the integrity of the sample. The sample is not stained. The only thing that we will be able to see with a confocal is some autofluorescence. I find that since we have that sample finder installed here on the LSM 900, I find myself playing around with different samples a lot more than I used to, because it's so much easier to get started. So the last 3 days, the last 2 days when I was preparing this, it was actually a lot of fun to go in with different samples that I normally don't try to look at often. With a confocal, but this is one of them that I thought was most fun, is because it is a large sample, and second, we can see some interesting details, and just happened that they are autofluorescent. So that was actually a quite nice surprise. So okay, we checked around the sample. We find that it is in focus, and yes, we can get some contrast-rich images. This is a 2.5x objective. We don't expect the details to be, you know, super fine. I can try to focus around here using the control button. We can move around. This is a very, very thin specimen, and the 2.5x objective actually has a very large depth of focus. So focusing around the sample with a 2.5x objective does not bring a lot of 3D structures. Okay, so at that point we say, well, okay, fine. Do the preview scan. It's now going to move to the different areas of the sample. It's going to collect a composite image. This is an overview image. You may notice that some areas are brighter than other areas. This is because every area is illuminated slightly different in a different part of the focus. The LED lamps are not exactly in sync with that overview image. It is supposed to do it quickly and not accurately, but we can generate after that. So if I press finish and I select an experiment, let's use, I don't know, the last one I used was a confocal kind of experiment that is preloaded into the sample, and that leads us directly to the scanning area. So here again, we can navigate to individual parts of the sample. For example, I would like to take a look at this area here. Sometimes with large samples such as these, it's easy to get lost. Like if I zoom out and I zoom out into a different section of the sample. Okay, so now I'm genuinely lost. There is an important button that I would like to show you. That is that center to stage position. That's whenever you're lost, it sends you back to wherever the stage is. It's an important new button that I'm not sure we have it on the previous instances of the LSM 900, 980, and so forth. So that's a nice little button to have. So in this area, I don't want to measure it with a 2.5x objective. I actually want to go in more detail. I want to use the 20x objective. So I'm going to click on the 20x objective. And you see now here, the area I'm looking at is a lot smaller. And okay, I'm zoomed in with a factor 3.2. I'm going to zoom out again. I want to catch one of these round objects here. I'm going to move that region of interest to one of these areas down here. Okay. And then I would like to collect images over there. So when I start this continuous scan here, I will probably not be perfectly in focus. That's one of the things about the AI sample finder. It is not going to give you the perfect focus right away. So that's where we would start to go. Oops, what happened here? That's where we would start to go. And why is everything suddenly so bright? Yes. Okay, that's where I would then click here again and try to focus this a little bit better. I'm moving sample up and down. I should be able to see some kind of background haze. Okay, let me start again, maybe with a little bit more laser power on all the channels. The pinhole is actually quite open. Ah, yeah, the gain was quite low here everywhere. So the sample is, we're only seeing it by virtue of its autofluorescence. So we're not really going to okay, so now at least we see some noise. Yeah, there it is, the noise. And here I can start to move up. And down into the sample and hope to see something. It's actually quite hard to see it here on my screen. It's actually quite hard to see here on my screen. Okay. I think I'm not focused into the sample. I think what I see there is some kind of a surface moving up a little bit more. Yeah, that's where I'm starting to see more signal. Okay. Okay, now we're talking, huh? I'm starting to get more and more signal here. Yeah, and there's the structure. That, yeah, now that's more like it. I was just at the wrong place. Okay, so that's the fluorescence image at that area, and I can move here around and see these different areas. And then I can go back to the, so if that's what I defined, I can go back. I could, in principle, start the experiment, and then it would collect the entire animal with that. But in the interest of time, I'm going to remove that area of the tiles, and I'm going to generate a different one. I'm just going to collect a smaller area around here, and then do the experiment over there. Oh, I think I had the autofocus on. Yeah, my bad. I'm going back into this continuous thing. I'm going to tell it, okay, so this is the focal area I want to use every time. Yeah, the intensity here was too high because when I was looking, I increased the laser power too much. I'm going to reduce that a little bit. Okay, there we are. And then I'm going to switch off that focusing thing for the time being. And then collect the confocal image over that area there. So okay, that's how that thing works. If we were interested in collecting a different area, for example, there on the back, if I go over here, let's see how that thing looks like. Oh wait, let me recall the focus. Okay. That's that area on the back. And with that, I can navigate back and forth. Okay, I have some latency issues here on my computer. I'm not sure if you have that over there on your stations as well. If I go use this, go back to the center stage position. Oh, now it went back to that first position where we were. Oy. But actually we want to go, yeah, latency is really a problem. And I would like to go into this area over here. Yeah. And I want to take a look at this area. and collected object. So yeah, there we go. So sample finder helping navigate. What I'm going to do now is I'm going to stand up here from my chair. I'm going to rush over to the LSM 980. I'm going to swap samples. I'm going to put in a sample with a typical cell culture, right? And see how the sample finder works on that scenario. Bear with me. I'm going to mute myself and I'll be back in like 2 minutes. Okay, I'm back. Hi, can you still see the screen?
Yes.
Okay, so I just placed the sample in and I'm going to press now the find sample button. It is going to collect an overview image of that specimen. It's just a cover slide with some HeLa cells on it. Okay, it should now have no problem finding the cover slip. And since there is no specific object in there, there is no tissue cut or anything like that, it's not going to find any sample. Yeah, it has not found any sample, but that's not a problem. We're going to tell it, okay, the sample is somewhere in the middle here. Also, that process should get us to the image rather quickly. Okay. So again, if I use the coherence contrast imaging on this area here, it is not going to resolve every little detail of the cells, but at least it should get us some kind of an idea what the sample might look like. So if I go to live, yeah, if I zoom in here, we see all these little dots here. These are, well, you can almost not see them from far away. But if I zoom in over here, we can see individual dots. These are the nuclei of the cells that are spread around. So yeah, it's right there where it's supposed to be. I could look at different areas of the sample. It's actually a quite homogeneous one. Maybe at the corner of the, maybe here we can see here where the mounting medium stops and all the imperfections of the glass there at the corner. Okay, so we suppose we want to collect an image right at this place here. Then we're fine. We say, okay, stop this thing. I don't need a preview scan right here. Well, okay, let's do a preview scan. Why not? It will collect the images there. And I say here, okay, finish. And then it's going to ask me for some kind of a confocal setting. I'm going to use the same as we had before, why not? Again, I have no idea what the sample is, but I could also turn on a green channel over here and do it in a line-wise switching, turn on all these colors, okay, over that area here. I'm going to select an objective with a higher magnification, go into that little lump of cells over here and hope to see some of them appear. So if I hit continuous, it should get us, yeah, it's not exactly in focus, but it's getting there. Yeah. Okay, so that's where the cells are. Here I would do the usual adjustments. Maybe the 405 light is too strong. I can go with a milder laser here in the DAPI channel. I can also do everything milder here, probably at 0.5 everywhere. There is also, I think the green channel is laminin enveloping the nucleus. So I'm going to turn everything down. Yeah, it's probably even more. We can now at this point go with a lower gain on these channels. So I set up the image just like I would usually do it. It'll look somehow like this. Okay, we still have very strong nuclear staining. We still have very strong red staining. We still have very strong also far red, and the laminin here. Yeah, we really can go down with the 488 power. Oh, yeah, here, by the way, I'm using the wrong laser. Can change it. Okay, so now we see more or less the structures of these cells like we need to see them. And then if I would be interested to count how many cells are in there, I would again, use the definite focus and say, okay, this is the focus that I would like to use. Go to the focusing strategy and say, okay, you know what, use the definite focus, do it every tile at this position that that we were defining over there. And yeah, let's do these tiles over there. The other thing that I would like to point out at this point is we can go to individual positions of the tile, and we could actually define where we want the autofocus to take place. So if we do here focus, surface, and support points, and we could turn the focus strategy to say, okay, use whatever is defined in tile setup. Then we can set it up here in tiles, and we say, okay, I would like to have 3 columns and 3 rows worth of focus points. These focus points appear here. And it's not very well visible here on the screen, I think, but here is one of them. And I can move it. If I have structures that are out of focus, or if I have a horseshoe-like specimen, where I want to make sure that the focus is really taking place within the area that I'm interested in, I can define these focal points there. And I can go also to each and one of these points and ask to verify these points. So I'm going to go and tell it, okay, I want to make sure that we are actually measuring these points here in focus. So I'm going to recall the focus here using the definite focus. Okay, I'm going to use this definite focus as a device to recall this thing. And they say, well, okay, this one is in focus. I set this as my Z position, move to the next. Then I want to run this definite focus thing again. Okay, it finds that. Move to the next. Yeah, that's fine. And then I want to use this definite focus to verify all the positions. So it's rewriting here the numbers from 1200 something to 1377. And that actually goes along the warp of the cover slide at that point. And once that is done, I can start the experiment. And now it's going to collect the confocal images over the entire area there. Here we have some events of cells dividing. We have some events of the cells nicely spread. And this whole experiment will take around 5 minutes. Here we have many cell divisions, many different types of cell division here. That would be a nice example to take a look at in the IntelliSys, the image segmentation, to find out, the conditions of all nuclei and to count the entire area. Yeah, that's how you would go about collecting images of a single cell layer like cell culture with the help of the navigation and the AI sample finder. So the AI sample finder in this case, what it did is it primed the image acquisition so we can see something, so we can navigate easier in the navigation setup. In the other instruments that do not have the AI sample finder, it is also possible to go in with a zoomed out kind of setting and a quick scan with very low resolution and set up a preview scan of the area of interest. However, since we start from scratch, you don't even know where the sample is. So simply by having like a webcam-like image of the sample where it is about, and having that encoded with the right positions on the stage is a tremendous help to get started to collect these images. Yeah, I hope that this demonstrated it well, what can be done with the help of the AI sample finder. Yeah, while this image is acquiring to the end, I would like to open the chat and ask the participants if there are any questions. That would be a nice time to ask questions. We can also, I have more samples in there. If you want, we can run a different sample. Just let me open the channels for the communication at this point.